In this session, we will discuss about compound microscope. Compound microscope is different from simple microscope because it is made up of combination of more than two or more lenses. Compound microscope. The compound microscope is not a simple microscope because here in compound microscope, combination of lens are used, and ocular and objective lens are used. And this objective and ocular lens will magnify the image, and we can observe or see the enlarged image with clear picture. And these are the parts of a compound microscope: ocular lens, body tube, arm of the microscope, nose piece. The nose piece, you can see the objectives. This is the stage, and this is the clip. This is the condenser, diaphragm, light source, coarse adjustment, fine adjustment, and base. And these are the parts of compound microscope. We will go in detail about that. This is body tube, and this is the body tube. The body tube holds the objective lens and the ocular lens at proper distance. And here is the nose piece. The nose piece holds the objective lens and can be turned into increase the magnification. And this objective lens is connected in the nose piece. By rotating the nose piece, we can select the different magnification of objective lens. And this is the ocular lens. And through ocular lens, we can see the enlarged view of the object or the sample. And this is the arm of microscope. It is used to support the microscope and carry it, and also hold the body tube, nose piece, and objective lens. Objective lens. The objective lens increase magnification of the image. In microscope, there are two types of lens are used. First one is objective lens, and this is the ocular lens or eyepiece. So when the light rays passes through the specimen, it will enters into the objective lens. So the objective lens will magnify the image. In compound microscope, we used different type of objective lens depending upon the magnification what we need. And in microscope here we use 4x, 10x, 20x, 40x, and 100x. And this objective lens is present in the nose piece. And by rotating the nose piece, we can change the magnification of objective lens. And this is the stage. The stage will support the slide. And here in the stage, we keep the specimen. Stage clip. These are two stage clip hold the slide or specimen in place on the stage. Then simply the stage clip will hold the slide. The condenser. The condenser focus the light and illuminate the sample. This is the condenser. When light passes through the condenser, condenser will focus the light into the sample. Also, it will illuminate the sample. So we will get a clear or better image. Diaphragm. This is the diaphragm. The diaphragm control the amount of light on the slide or specimen, or also we can say that the diaphragm will control the intensity of light to the specimen. Coarse adjustment. Move the body tube up and down for focusing the image. So that means the coarse adjustment will help us to focus the image. Fine adjustment. This is the fine adjustment. This knob moves the body tube slightly to sharpen the image. That means first, with the help of coarse adjustment, we will focus the image, and with the fine adjustment, we will sharpen the image for clear visibility. Light source. Project light upward through the diaphragm, the specimen, and the lens. And this mirror and electric bulb are used as light source in earlier times, or in the basic simple microscope we used mirror as light source. So the light will come and hit this mirror. The mirror will reflect the light towards the specimen through condenser. And nowadays in most of the microscope we used electric bulb as light source. At last, this is the base which support the microscope. So the principle of compound microscope, and these are the parts of compound microscope. This is the light source, and this is the condenser. In between condenser and light source, there is a diaphragm is there, and this is the specimen stage. We will keep the this microscope stage. Here we will keep the specimen, and these are the objective lens, and these are the ocular lens. So first, the light will passes through the diaphragm. The diaphragm will control the light, or diaphragm is having a shutter. If you close the shutter, the light will not passes through this diaphragm. It will not reaches to the condenser. So by adjusting the shutter of the diaphragm, we can control the light. So the light will reach us to the condenser. So what will happen? This condenser will focus the light to the specimen, and also it will illuminate the specimen or the object. Okay, and after that the light will reach us here, the objective lens. So the objective lens will magnify the image. There are different type of objective lens are there. We already discussed earlier. 40x, 4x, 10x, and 100x. Depending upon the need, we can change the power of this objective lens. Or simply we can say that this objective lens will multiply or magnify the image. When this light rays passes here, reaches the ocular lens, or this ocular lens will also magnify the image. And from here, with the help of our eyes, we can see the enlarged view of the microorganism or the object. Magnification. Magnification is also called as the power of microscope or enlargement of microscope. We will call it as a magnification. In microscope, we already told that the tiny object or microorganism is magnified for better observation. In compound microscope, there are two type of lens are used. One is this is the objective lens and the ocular lens, or you can say that this ocular and this objective lens will enhance or magnify the image. And the total magnification of the microscope can be calculated by multiplying the power of ocular lens and objective lens. And in compound microscope, there are single type of ocular lens is there, mainly that is in 10x, and different type of objective lens are used, that is 4x, 10x, 40x, and 100x. And this lens can be changed depending upon the need of our observation. So in magnification power of low power is 100x. In low power, we can observe the protozoa and larger microorganism. And this magnification power of high power is 400x. With the help of this high power, we can observe the motility of microorganism, mainly motility of bacteria, protozoa, and fungi. In oil immersion, the maximum magnification is 1000x. With the help of this oil immersion microscope, we can stain the smeared, or we can observe the stained smear. Oil immersion. In oil immersion of compound microscope will give high magnification power up to 1000x. Oil immersion we use to observe stained bacterial smear. Not only the bacterial smear, stained smear we can observe in oil immersion microscope or oil immersion objective. This will give a clear image. In oil immersion, we place an oil between the smear and this objective lens. By that, we will get a clear image of the organism. We can study the morphology of stained smear very clearly, because oil will fill the gap between the smear and objective lens. Because of the same refractive index of oil will avoid the refraction of light. That is, all the light will passes through the smear will reaches the objective lens. So we'll get a clear image of the magnified picture. So this is the oil immersion light. This is the light source. The light source will pass us through this oil. While passing, what will happen? The scattering of the light or the refraction of light will be limited or controlled. So all the light will reaches to the objective. So we'll get a better image. And here you can see, due to the absence of this oil, the light will be refracted or scattered, and only some amount of light will reaches to the objective lens.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.